So in section 8.4, we're looking at nomenclature and classification of organisms. Nomenclature actually refers to the system of naming that we use. We now use something known as binomial nomenclature that you guys have probably remember from Bio 1. But the system of naming is based on how we classify organisms and what taxa do we put those in. So, first of all, how do we group or classify living organisms today? Remember, when we talk about living organisms, we could be talking again about organisms from the past that are now extinct. Or we can also be talking about organisms that are currently living, which are known as extant.
Kathy Hardin, please call the front office. Kathy Hardin, please call the front office.
Now, there are multiple correct names for an organism, and oftentimes these correct names are referred to as the common name. The common name is going to be based on the spoken language and is often used in colloquial language. Okay, common everyday language. A common example that you'll see for common names is going to be your water moccasin versus your cottonmouth snake. Both of these names actually refer to the same exact snake, except one of them, the water moccasin, refers to the fact that it is actually a semi-aquatic snake. Can actually live and swim in the water. And on the other hand, the cottonmouth actually refers to the white of its mouth and how it actually looks right before it attacks or right before it strikes. Other times, depending on the region in which you live, the two common names can differ as well. So in some places they might notice more of them living in that aquatic region, and others maybe on land, but we've noticed how they look before they attacked or what are their specific characteristics. However, when you look at a scientific name, this is going to give us more information. And the scientific name is based on the Latin language. And the system we use is known as binomial nomenclature. This binomial nomenclature was actually developed by Linnaeus, and with the Latin language, this is intended to provide us with more accurate information on the organism. Info on organism. So, an example, with the same snake, the water moccasin, or the cottonmouth, its Latin name or its scientific name is Agkistrodon piscivorus. And usually this name, if we were writing it out, we'd actually underline it. These scientific names will also show relationships, so relating it back to our unit on phylogeny, on our section on phylogeny. When you have the scientific name is going to be broken into two parts. Binomial nomenclature, two parts. The two parts are going to be known as the genus and the species. Notice how I'm writing that. You have to actually italicize this or underline if you're handwriting it. The genus is always capitalized, the species is always lowercase. An example, your domesticated dog is known as Canis familiaris. And again, since I'm handwriting it, I will underline it. You should be able to identify that. So how do we actually find a scientific name? Finding a scientific name could be done through the use of what's known as a dichotomous key. In a dichotomous key, di means two, or two different, actually uses or is a series of questions that gives at least two different options. So for example, this is a kind of a simplified dichotomous key, more of a picture than a word key. You're going to start with your first question, and the first question says, do you have feathers? Yes or no? If you have feathers, you go to, oh, does it swim? Yes or no? If it does swim, then yes, you're going to be a duck. If it does not swim, then no, you're going to be a hen. Obviously a very simplified image of a dichotomous key. But other times you can see a dichotomous key. An example of a dichotomous key would be a field guide. For example, for snakes, birds, plants. They will often provide you with physical characteristics for you to choose from, and then that gives you, could be the common name of the organism, or sometimes the scientific name as well. So just keep in mind that the idea of our scientific names is that it's going to be much more accurate, and especially when scientists are communicating with each other from different countries, this does simplify that process as well. When you are using something like a dichotomous key, you want to make sure you always start at the beginning, even if you're looking at 5 different organisms. And, of course, this is only going to be helpful if we already know the names of those organisms. But moving along with our short lecture on classification is looking at the history of classification. At first, we had Aristotle. And Aristotle first divided all living things into either plants or animals. Later on, we had Carl Linnaeus. And Linnaeus actually created taxons, or these levels of naming, which eventually led to our binomial nomenclature, our scientific name. Excuse me. So if you're looking at this image here, our taxons are described here as kingdom, phylum, class, order, family, genus, and species. And the genus and the species is going to produce our scientific names. In this image, we start with the animal kingdom, and in the animal kingdom we have a wide variety of different animals here. But as we break them down into phylum, we're going to describe some specific characteristics. So, what is our current system of taxonomy? Well, with each taxon describes a specific characteristic. And on this image, we're actually missing one of our classifications, which is known as the domain. The domain is sometimes seen as a separate grouping, but the domain really breaks our living organisms into 3 key groups. That's going to be our eubacteria, archaebacteria, and our eukarya. After that, we have our kingdom, phylum, class, order, family, genus, and species. Okay. The domain or the kingdom are going to be what's the word that we use is inclusive, because it includes many living organisms. It can include all eubacteria, it can include all archaebacteria, or it can include all eukarya, which means our eukaryotic organisms. Just keep in mind that our eubacteria and archaebacteria are prokaryotic for right now. And of course, our eukarya are our eukaryotic cells. You should be able to memorize the order of this, and often a common sentence that you'll hear to help remind you of this is that deer, and again, sometimes the domain isn't included with these taxons on their own, just like in the image, but this is a grouping that you should not forget. It could be deer, King Philip came over for green soup, is what I learned. I've also heard grape soda. Okay, but this can help you remind you of the order of things. Domain and kingdom being the most inclusive groups, and species being the most exclusive. So if you notice up in the picture, you have all of these different varieties of animals, but as you move down towards species, you have one specific species, and that's going to be very exclusive to that group, that naming. Scientific name, again, is an example of our binomial nomenclature. And naming in general, taxonomy, actually needs to be flexible. This field of science is very flexible as we learn more info to accurately describe organisms. So we've learned more and more, and our grouping of organisms has significantly changed due to that. Other information, we want to very quickly kind of move through the rest of this chart here. Here we have the description of the 6 kingdoms, and again, you just want to keep in mind that we also have 3 domains related to these kingdoms. Archaea is going to be related to kingdom Archaebacteria. Our eubacteria is related to our bacteria kingdom. And then our eukarya is going to be related to the rest: protists, fungi, plants, and animals. In this section of notes, or in this chapter, we'll actually be focusing on primarily the protists and the fungi, but you do need to remember some general information on our animal cells, or about our animals and other cells as well, other cell types. But with archaebacteria, things you have to know is that they live in very extreme environments and are known as our extremophiles. However, our bacteria, or eubacteria, are our common bacteria, what lives on, around, and maybe even in you all day, every day. As far as having a nuclear envelope, both of these do not have a nuclear envelope. However, they have what's known as a nucleoid region. Additionally, inside that region, or in that region, not inside of, you actually have circular DNA. In our eukarya, we actually have nuclear envelope in all of them, a nucleus, because they are all eukaryotic, okay? They're also going to have what's known as linear DNA. When we talk about protein synthesis and transcription and translation, this is going to happen simultaneously in both of these groups. And actually, the ribosomes can actually be smaller in our bacteria groups. Oops, I can't spell. Whereas in our eukarya, this is going to happen separately. And you should remember that transcription happens in the nucleus. and translation happens in the ribosome, or at the ribosome. When we talk about the cell wall, our archaebacteria does have a cell wall. You need to know that it's not made of cellulose, and it's also not made of the carbohydrate peptidoglycan. For our eubacteria, we do have a cell wall made of peptidoglycan. And excuse me, I think I said this was a carbohydrate. It's actually a type of protein. Cellulose is a carb. Peptidoglycan is a type of protein. In protist kingdom, yes, we do have some with a cell wall. In fungi, we have generally a chitinous cell wall, and again, not a cellulose cell wall. Plants, we also have a cell wall, but it's going to be cellulose and other polysaccharides. In the animal kingdom, we generally don't have a cell wall. However, some of our organisms, like our arthropods, like lobsters, could actually have a chitin shell. However, that's not really the same idea as a cell wall around each of their individual cells. For modes of nutrition, you need to read through this. One thing we should add is that for fungi, they have extracellular digestion. And the other thing we should add is for multicellularity, our bacteria, of course, are unicellular, and you want to keep in mind that they are generally about 1/10 the size of our eukaryotes. And that's all for today. Make sure you read through the rest.